A young man is driving through the Four Corners region of New Mexico on the 14th of May, 1993. He is 19 years old. He is a marathon runner. He has no medical history worth mentioning. He starts having trouble breathing. He pulls over. He cannot get enough air. He makes it to a hospital. He dies within hours. The cause of death is listed as acute respiratory failure of unknown origin. But the detail that makes the attending physicians stop and look at each other is not the death itself. It is the destination. The young man had been driving to a funeral. His fiancée had died 5 days earlier. By May 17th, physicians at a medical center in New Mexico have identified 3 more deaths. Same region, same presentation. All previously healthy adults. All dead within days of their first symptom. Something is moving through the Four Corners. Nobody knows what it is. Nobody has seen it before. And nobody, anywhere in the Western Hemisphere, has ever named it. The Four Corners region is where the borders of New Mexico, Arizona, Colorado, and Utah converge. It is high desert plateau, red sandstone mesas, juniper woodland, and open sky. The Navajo Nation covers a significant portion of it. It is one of the most geographically remote regions of the continental United States. In the spring of 1993, it is also producing one of the most unusual ecological conditions anyone in the region has observed in decades. The winter of 1992 and 1993 was abnormally wet. An El Niño event had pushed moisture into the southwest. The desert had responded in a way it rarely gets the chance to. Pinyon pine nut crops were extraordinary. Grass and shrub growth was exceptional. Food was everywhere. And where food is everywhere, mice multiply. The deer mouse, Peromyscus maniculatus, is a small brown rodent found across the North American continent. It lives in grasslands, scrublands, forests, and desert edges. It shares its range with humans everywhere in the American West. In normal years, the deer mouse population in the Four Corners region is stable and unnoticed. In the spring of 1993, biologists from the University of New Mexico, who happened to be conducting a population study in the region, made a measurement that stopped them. The deer mouse population had increased tenfold compared to the previous spring. 10 times as many mice, moving into barns and outbuildings, into storage sheds, into corners of houses, into the spaces where people live and work and breathe. And each one of those mice was carrying something. It had always been there, living silently in the rodent population. But until May of 1993, nobody in medicine had ever seen it reach a human being in the Americas. On the 18th of May, the New Mexico Department of Health contacted the Centers for Disease Control and Prevention in Atlanta. The message was simple. We have a cluster of unexplained respiratory deaths among previously healthy adults. We need help. Within hours of that call, a team was assembling. Jay Butler, an experienced epidemiologist in the Epidemic Intelligence Service at the CDC, was designated as team leader. 2 young Epidemic Intelligence Service officers, Ronald Moulinar and Jeffrey Dushan, were assigned to assist him. Less than 24 hours after the team was organized, they arrived at Albuquerque Airport. They were met by members of the University of New Mexico medical faculty, Indian Health Service physicians, and state and federal health officials. The first order of business was a case definition. Every investigation needs a starting point, a description of what they are looking for. The team agreed to evaluate any patient from the area who, from January 1st, 1993 forward, had demonstrated imaging evidence of unexplained bilateral lung infiltrates with associated hypoxemia. That definition would prove critical, because as they began looking, they found that cases had been occurring since March, weeks before anyone had connected them, weeks before anyone had seen the pattern. The clinical presentation was consistent across every patient. It began with fever, muscle aches severe enough to make movement difficult, headache, fatigue. These initial symptoms looked exactly like influenza. They looked like 100 other things that physicians see in the spring. But then something happened that influenza does not do. Within 2 to 4 days of the first symptoms, the lungs began to fill with fluid. Not gradually, rapidly. The medical term is non-cardiogenic pulmonary edema. The practical reality is that the lungs stop working faster than any treatment can respond. Blood pressure dropped. The heart failed to compensate. Without aggressive intensive care intervention, patients died within 24 to 48 hours of the pulmonary phase beginning. In the early cluster, the case fatality rate was 52%. More than half the people who got it died, and the physicians treating them had never seen anything like it. The investigators began ruling out everything they knew. It was not influenza. The clinical course was wrong. The severity was wrong, and the laboratory findings were wrong. It was not pneumococcal pneumonia. It was not Legionella. It was not hantavirus, as it was known. That last exclusion was significant. Hantaviruses had been known to medical science since American soldiers developed a hemorrhagic fever during the Korean War in the early 1950s. The disease they developed was called Korean hemorrhagic fever, later named hemorrhagic fever with renal syndrome. It primarily affected the kidneys. 3 strains of hantavirus were well documented by 1993. Hantan virus from Korea, Seoul virus from Asia, Pumala virus from Northern Europe. All 3 caused kidney disease. None of them caused lung disease. None of them had ever been associated with a case in the Americas. But when the Special Pathogens Branch at the CDC tested extracted antibodies from 9 of the patients against a panel of 25 known viruses, something remarkable happened. Every one of the 9 samples reacted to all 3 known hantavirus strains. None of them reacted to any of the other 22. The investigators had found their starting point. But it was a hantavirus, unlike anything anyone had described before. And it was in a place no hantavirus had ever been found in a human being. On the 4th of June, the hantavirus hypothesis was strong enough to drive the next phase of the investigation. A rodent trapping operation was launched across the Four Corners region. From the 7th of June through mid-August, the field teams captured approximately 1,700 rodents from patient sites and control sites across New Mexico and surrounding areas. The most commonly captured species was the western deer mouse. At the same time, scientists at the CDC Special Pathogens Branch in Atlanta were racing to identify the virus at the molecular level. On the 10th of June, using reverse transcription polymerase chain reaction technology, a research team obtained a DNA sequence 139 base pairs in length from one segment of the virus genome. The samples had come from 2 people who had died from the disease. It was a hantavirus, but it was clearly a new strain, a strain never previously identified, never named, never described in any medical literature. Less than 1 week later, on the 16th of June, the same team identified an identical viral base pair sequence and hantavirus antibodies in deer mice trapped at the locations where patients had lived. The virus in its reservoir had been identified. In less than 3 weeks from the assembly of the task force, the CDC had found what it was looking for. It was one of the fastest pathogen identifications in the history of American medicine. The mechanism of transmission was traced through the case control study. Investigators compared the households of patients with nearby control households. What they found was that patient households had significantly more rodent activity than control households, and the patients were more likely to have recently engaged in activities that disturbed dried rodent material, sweeping barns, cleaning storage areas, turning soil for planting, opening outbuildings that had been closed through the winter. All of these activities had one thing in common. They disturbed dried rodent droppings, urine, and saliva. They turned solid material into airborne particles, and someone breathed in what was in the air. The virus traveled from the rodent to the human not through contact, not through a bite, but through the air itself, through the simple act of breathing. The virus targeted the pulmonary capillary endothelium, the cells lining the tiny blood vessels deep inside the lungs. It caused those vessels to become permeable. Fluid leaked from the blood into the lung tissue. The lungs began to drown from the inside. By late June, testing results showed that approximately 30% of the deer mice trapped across the Four Corners region were infected with the virus. And late that summer came a finding that changed the fear profile of the outbreak entirely. The virus did not spread between people. It could not move from one human being to another. Every single case required exposure to rodent material. That finding transformed the public health response. The outbreak was not a spreading infection moving person to person through communities. It was a point source exposure, requiring a specific action in a specific environment. Knowing that changed everything about how to protect people. Naming the virus was harder than finding it. The initial proposal was Muerto Canyon virus, after a location near where the first cases had been identified. But the Navajo Nation Council voted unanimously to oppose that name. The canyon called Muerto Canyon takes its name from a massacre of 115 Navajo people by Spanish troops in 1805. Associating the new disease with that name added an additional layer of historical weight that the Navajo community found deeply objectionable. A second proposal was Four Corners virus. That name was also rejected, this time by the communities of the region who feared continued stigma. The media coverage of the outbreak had already caused significant damage. Tabloids had called it the Navajo disease. Vacation reservations across the region had been canceled. Major events had been moved. Navajo students at some universities had been required to undergo medical testing before being allowed on campus. The economic and social damage to communities that had no more exposure risk than anyone else in the region was severe and unjust. The virus was ultimately named sin nombre, Spanish for without a name. The disease it causes is hantavirus pulmonary syndrome, the name chosen to protect communities that had already suffered enough. The Navajo Nation had known about cycles like this long before 1993. When CDC investigators met with tribal elders and Navajo healers, they were told something that the scientific literature had not recorded. The elders described previous outbreaks of similar illness. They had occurred in association with specific ecological conditions: years of excess rain, abundant pinyon nuts, increased rodent populations, cycles that the oral record of the Navajo people had documented across multiple generations of the 20th century. In Navajo tradition, mice and humans come from different worlds and are meant to be kept separate. The elders taught that contact with rodent waste could bring disease. They recommended burning contaminated clothing, sealing food against rodent access, careful handling of any space where mice had been living. This knowledge was not new in 1993. It was ancient. The epidemiological investigation confirmed exactly what the traditional knowledge had preserved. The virus required rodent material to reach a human being. The behaviors the elders warned against were precisely the behaviors that carried the highest risk. The connection between indigenous knowledge and laboratory science in the Four Corners investigation became one of the defining features of how the outbreak was ultimately understood. The investigation also reached backward into the past. Researchers working with archived tissue samples discovered that sin nombre virus had not appeared in 1993. It had been circulating silently for decades. The earliest confirmed case of hantavirus pulmonary syndrome in the United States was a 38-year-old man in Utah who died in 1959, 34 years before anyone in Western medicine had a name for the disease that killed him. Between 1959 and 1993, isolated cases had likely occurred across the American West. They had been filed as unknown respiratory illness, moved on from not connected. The ecological conditions of 1993, the tenfold increase in deer mouse population, the resulting explosion in human contact with infected rodent material, had produced a cluster large enough to be recognized. The virus had not emerged in 1993. It had been invisible until 1993. The total death toll from the 1993 outbreak was 33 people across the Four Corners states. Nationwide that year, 27 of 48 confirmed cases resulted in death, a case fatality rate of 56%. What the outbreak produced in the years that followed was substantial. Hantavirus pulmonary syndrome became a nationally reportable disease in the United States. Every case had to be documented and reported through the public health chain. That reporting requirement created a surveillance system that had not previously existed. The CDC and state health departments developed standardized protocols for investigating suspected cases. Laboratory diagnostic capacity was built and distributed to state public health laboratories. The investigation methodology developed in the Four Corners became the template for subsequent outbreak investigations, not only in the United States, but across the Americas. New hantavirus strains were identified in subsequent years. Bayou virus in Louisiana, Black Creek Canal virus in Florida, New York virus, Andes virus in South America, which uniquely showed evidence of limited person-to-person transmission. In each case, the framework developed in 1993 provided the starting point for the investigation. The surveillance system that emerged from the Four Corners has been directly credited with faster identification of subsequent outbreaks, which allowed faster containment and fewer deaths. In 2012, a cluster of cases appeared among visitors to Yosemite National Park in California. 10 people were infected, 3 died. But the cluster was identified within days, not weeks. The National Park Service issued immediate warnings. Thousands of potentially exposed visitors were notified. The speed of that response was measurable and direct. It came from infrastructure that did not exist before 1993. What changed for the people who lived through the outbreak and its aftermath was also substantial. The Navajo Nation implemented long-term prevention and control programs addressing rodent populations and surveillance. Public health education was developed and delivered in ways that respected and incorporated traditional knowledge rather than replacing it. The CDC Foundation worked with the Navajo community to implement the practical prevention measures that the investigation had confirmed as effective. Keep food sealed. Reduce rodent habitat around homes. When cleaning spaces where rodents have been, wet the area first before sweeping. Do not create dust. Ventilate. These practices, simple and actionable, came directly from tracing the transmission pathway from mouse to human and writing it down in language that anyone could use. The investigators, who did not sleep for 3 weeks in the summer of 1993, produced something that outlasted the outbreak itself. They produced the answer. And the answer is still working. The deer mouse is still in the Four Corners. Sin nombre virus is still in the deer mouse. In most years, in most places, the ecological balance holds. Rodent populations stay within normal ranges. Human exposure stays below the threshold where cases cluster. But the ecological conditions that produced the 1993 outbreak are not unique. They can occur again when rainfall is heavy and vegetation is abundant, and rodent populations respond to available food. The monitoring systems built after 1993 exist precisely to detect those conditions before they produce another cluster. The reporting systems catch individual cases that would previously have been filed as unknown respiratory illness and moved on from. The laboratory capacity allows rapid identification of any new hantavirus strain that the deer, mouse, or any other rodent might carry. Between 1993 and 2013, the CDC recorded more than 600 confirmed cases of hantavirus pulmonary syndrome in the United States. The case fatality rate remained near 35%. That number is high. There is still no specific antiviral treatment for hantavirus pulmonary syndrome. Survival depends on early recognition, aggressive supportive care, and sometimes extracorporeal membrane oxygenation. But the number of cases caught early, the number of clusters identified before they became the scale of 1993, the number of communities warned before the ecological conditions tipped into outbreak conditions is not visible in the mortality data. It is visible in the absence of another 1993. The young man who died on a highway in New Mexico on May 14, 1993, had no idea what was in the air of the region he was crossing. Neither did his fiancée. Neither did the 34 others who became cases that year. But the physicians who noticed the pattern, the state officials who made the call, and the investigators who assembled within 24 hours and did not stop until they had found what they were looking for, built something from those deaths that had no name. They built the name, and everything that came from the name, the surveillance, the protocols, the prevention guidance, the international templates, is still protecting people in every state where a deer mouse runs, in every barn that gets cleaned in the spring, in every country in the Americas where a small brown rodent carries something invisible, sin nombre, without a name. It has one now. And so does the system that protects the next valley, the next community, the next cluster that has not happened yet. The virus was always there. The investigation that found it built something that will be there longer. The investigation that identified sin nombre virus in 1993 was not the product of a single insight or a single institution. It was the product of a network that assembled because people made phone calls. The Indian Health Service physicians who were seeing the patients on the Navajo Nation in May of 1993 were the first layer. They had a unified system of interconsulting physicians, which meant that when unusual cases appeared across a wide rural geographic area, they could see the pattern. A physician treating a patient in one part of the reservation could connect their case to a physician treating a different patient 200 miles away. That network existed. It worked. And it produced the call to the New Mexico Department of Health. The New Mexico Department of Health made the call to the CDC on May 18th. That call was the second layer. The CDC assembled a team within hours and had investigators on the ground in Albuquerque within 24 hours. That response time was possible because the infrastructure for it existed. The University of New Mexico researchers happened to be conducting a deer mouse population study in the Four Corners region at exactly the moment when that data was most needed. That was not planned, but their data, showing the tenfold population increase, gave the ecological framework that made the rest of the investigation coherent. And the Navajo elders who told the investigators about previous cycles, who shared what the oral tradition had preserved across generations, contributed the historical dimension that no archive held. The investigation was a collaboration built from pieces that happened to be in place. Each piece was essential. None of them alone would have been sufficient. The publication of the hantavirus study group's research in the New England Journal of Medicine on the 7th of April, 1994, changed how the medical community understood the threat landscape of emerging infectious diseases in North America. The editors ran a glowing commentary alongside the research. They described the investigation as a model of collaborative outbreak science. They highlighted the speed of identification. They noted the integration of laboratory science, epidemiology, ecology, and indigenous knowledge as the framework that made it possible. The paper established hantavirus pulmonary syndrome as a distinct clinical entity. It described the laboratory profile in enough detail that physicians anywhere could recognize it. It outlined the case definition that became the standard for subsequent surveillance. That publication was itself a form of infrastructure. It put the information where every physician in the United States could find it. It meant that the next patient who walked into an emergency room with fever, severe myalgia, and a history of possible rodent exposure had a physician who knew what to look for. Before April of 1994, no physician outside the investigation team had that knowledge in any usable form. After April of 1994, every physician in the country did. The ecology of the Four Corners region continues to produce the conditions that drive rodent population cycles. El Niño events continue to deliver periodic wet winters to the American Southwest. Pinyon nut crops continue to respond to precipitation with extraordinary abundance in good years. Deer-mouse populations continue to respond to food abundance with population explosions. The relationship between climate, vegetation, rodents, and human disease risk in the American Southwest is now understood well enough to model. NASA researchers have used satellite data to map vegetation changes across the region and correlate them with deer, mouse, population dynamics, and subsequent hantavirus case rates. The technology exists to identify high risk years before the cases appear. That predictive capability grew directly from the ecological understanding generated by the 1993 investigation. The biologists who were already studying deer-mouse populations in the Four Corners when the outbreak began were integrated into the investigation. Their data became a foundational element of the ecological explanation. Their methodology became the template for ongoing monitoring. Every year since 1993, the relationship between rainfall, vegetation, and rodent populations has been tracked in the Four Corners region. When the numbers rise toward the threshold that preceded the 1993 outbreak, the surveillance system activates at a higher level. The monitoring intensifies. The public health messaging becomes more visible. The system is not perfect. Cases still occur. People still die. The virus is still in the mice, and the mice are still in the landscape. But the cases are found faster. The clusters are identified before they become outbreaks. The communities at risk are warned before the threshold is crossed. That is what? 24 hours of response time in May of 1993 and 3 weeks of exhaustive investigation in June produced for every spring that came after. The young man was 19 years old when he died. He was driving to bury his fiancée. Their names were not widely reported. The early media coverage of the outbreak focused on the disease itself, on the mortality rate, on the geographic location, in ways that were sometimes sensational and sometimes harmful to the communities where the cases had occurred. The people who died in the 1993 outbreak were young adults living and working in the American Southwest. They were farming and ranching families. They were Navajo Nation members. They were people who swept barns in the spring and turned soil for planting and opened storage sheds and breathed the air that rose from surfaces where mice had been. They had no way of knowing what they were breathing. The knowledge that would have protected them did not exist in Western medicine. It existed in the oral tradition of the Navajo Nation. The elders knew. The tradition had preserved it. But it had not reached the people in the barns. It had not reached the people turning the soil, not in a form that connected what they were doing to what could happen to their lungs. The prevention guidance that now exists, written in clear language and distributed through public health channels, is the product of tracing that gap and closing it. It will not reach everyone. It has not ended hantavirus deaths, but it is there. And It is working. The virus was always in the landscape. What was not in the landscape until 1993 was the name for it, the system to find it, the protocol to contain it, and the knowledge to protect the people living in it. Now all of those things are there, built in 3 weeks, expanded over 30 years, still running. The jungle was always there. They just did not know what was living inside it until they looked, and what they found changed everything that came after. Not just for the Four Corners, for every outbreak investigation that followed, for every country in the Americas where the virus had been circulating without a name, for every physician who would now recognize the laboratory profile before the patient's lungs were already full. 3 weeks, 33 dead, and a surveillance system that has never stopped running. That is the honest accounting of what the 1993 Four Corners outbreak produced. The loss was real. The response was remarkable. And what was built from it is still there,